But today we're going to start talking about Mendelian genetics. So we mentioned a couple of concepts already, so we'll see those again. But we are just kind of going to jump right into it. Gregor Mendel, he was a monk. He liked to play with pea plants, and he looked at different characteristics of pea plants. And he came up with a lot of really good information, some rules. He noticed a lot of patterns in the colors, sizes of his plants, and the shape of the peas, the actual peas. And so he is sort of considered the father of modern genetics, even though he knew nothing about DNA, nothing about chromosomes or anything like that. He just, grew a bunch of different pea plants and crossed them, and he counted the offspring, and he learned that there were some reproducible patterns when it came to inheritance. But as we're going to learn in future lectures, nothing is as simple as what Mendel envisioned. It's way more complicated. So he did his experiments in the mid-1800s. So again, knew nothing about the nucleus and chromosomes and DNA and any of that good stuff, mitosis, meiosis, nope, right? Didn't know that stuff. But he did learn that when he made certain crosses, when he mated certain pea plants, he could predict what the offspring would look like based on his previous experiments. So a true breeding variety are plants that always produce the exact same offspring. And those offspring produce the exact same offspring, so they're true breeding. If you cross two true breeding strains, you get a hybrid. That's called a hybridization. Hybrids are not true breeding. We designate the true breeding parents as the P generation, or the parental generation. The F1 generation, or the first filial generation, is the first set of offspring, and they're called the F1, and they are hybrids. So when you cross two true breeding parents, you get F1 hybrids. If you cross two of the F1 hybrids, you get the F2 generation. You should keep in mind that if you're doing this with animals and you're crossing two F1s, you're doing a brother-sister mating, right, for animals. With plants, it doesn't matter, but with animals, there are some issues with that, right? Now, when it comes to hybrids, they're not true breeding. So if you take seeds from a hybrid plant and plant them, you won't get the exact same offspring. Some of them will look like the parent, but not all of them. When Mendel crossed his true breeding white and purple flowered pea plants, all the F1 hybrids were purple. So even though the original parents, one was purple and the other was white, and normally if you breed purple with purple, everything's purple, and you breed white with white, everything's white. But when you cross the white with the purple, you still got all purple. So the F1s were all purple. But when you cross two of those purple F1s, the offspring, some of them were purple, some of them were white, right? So we're talking about flower color here. So the original parents, one was purple, one was white. When you cross them, all the F1 offspring, they're all purple flowers. If you take two of the F1s and cross them the offspring, the F2 generation, some of them had purple flowers and some of them had white flowers. And so the ratio of the purple to white flowers was about 3-to-1. So there were 3 times as many purple flowered peas as there were white flowered peas. And that's a ratio we're going to see many times, a 3-to-1 ratio. So here we have our true breeding plants, right? If you mate all purple with purple, you always get purple. White with white, you always get white. But when you cross the purple with the white, they're all purple, right? The F1s are all purple. Even though you made it with a white flower, you don't see any of the white flower plants in the F1, but if you cross two of the F1s together, then we get 3 times as many purple as we do white, but now the white shows up. And here, just giving some example numbers, 705 purple plants and 224. If you do the math, that's roughly 3 to 1, right? There are 3 times as many purple as there are white. The numbers are not exact because there's a lot of random variables that happen. If you were to grow thousands of these pea plants, you get an almost perfect 3 to 1 ratio. But you need a lot of plants, a lot of offspring to really see these ratios, right? Because there's always random variation, random chance, random gametes coming together, right? So, but a roughly 3 to 1 ratio in the offspring, right? Does everyone see that? If you multiply 224 by 3, you get, 672, which is close to 705. So roughly 3 times as many purple as there are white flowers. So Mendel reasoned that only the purple flower factor, right? Because he didn't know about genes, he didn't know about alleles, right? He just reasoned that the purple flower factor was affecting the flower color in the F1 hybrids. So he called that purple flower color a dominant trait. And the white flower color he called a recessive trait. And so Mendel used the term heritable factors because, again, he didn't know about genes or DNA or chromosomes. Of course, we call these heritable factors genes today. And when you have a different variation of the same gene, what do you call that? Alleles, right? So it's the same gene, but you have different versions of that same gene. And we call the different versions alleles. So you have dominant alleles and recessive alleles. So Mendel developed a hypothesis to explain the 3 to 1 inheritance pattern he observed in the F2. And there are 4 related concepts that make up this model. The first concept is that alternate versions of genes account for variations in inherited characteristics or characters. For example, you have one gene for the flower color, but it could be the purple version of that gene or the white version of that gene. And of course, as we said, these different versions are called alleles. So there's a purple allele and a white allele. Each gene resides at a specific locus, and locus means location, on a specific chromosome. So you find the genes, all genes in the same place, on the same chromosome for a given species. So, and that's very important because the genes have to be located in the same exact position on homologous chromosomes, so that if crossing over occurs, you still have a copy of those genes. You just may now have different versions of them. You may have different alleles of them because the adjacent sister chromatids, broke off and reattached. So you have now increased diversity, you've increased variation in those gametes, right, by mixing up all of the different alleles that you have. So just an example, here we have a homologous pair of chromosomes. These are not duplicated chromosomes, they're not replicated. We're just showing, right, the chromosome that came from one parent and the chromosome from the other parent. And so we have a gene for flower color in the same place on both chromosomes. You just have different versions of that same gene, so you have different alleles. Some traits just have 2 alleles. Other traits have multiple alleles. The second concept is that each character, each characteristic, each trait, an organism inherits two alleles, one from each parent. So again, we're using the word allele here. Mendel would have said heritable factor, but we do know that we have two copies of each gene. One comes from mom, one comes from dad. So you have two alleles, one from each parent that produced that offspring. And Mendel made this deduction without knowing about chromosomes or DNA. He observed his heritable factors, and he observed very characteristic ratios, very characteristic patterns in which those heritable factors were inherited. So the two alleles at a locus on a chromosome may be identical, as in the case of true breeding plants. So, right, the purple flowered plants, they have both alleles for purple. And the white flowered plants, they have both alleles for white. The difference is one of those alleles is dominant, right, the purple allele, and the other one, the white allele, is recessive. So the two alleles at a locus may differ, as in the case of the F1 hybrids. The F1 hybrids have a purple allele and a white allele, right, because they're hybrids. The third concept is that if two alleles at a locus differ, then one of them is the dominant allele, and that determines the organism's appearance, and the other is the recessive allele. It has no noticeable effect on the appearance. But that's if you have a hybrid characteristic where one allele is dominant and the other is recessive. You only see the dominant, not the recessive. So how do you see the recessive characteristic then? How do you see recessive traits?
In later generations.
I mean, but what do you have to have genetically in order to see them? Right, when both alleles are recessive. So the only way to see the recessive characteristic is that both alleles have to be the recessive allele. And then you see it. And what's the big word? If both alleles are the same?
Homozygous.
Homozygous. And if they're two different alleles?
Heterozygous.
Heterozygous, right? So in the flower color example, the F1 plants have purple flowers because they had the dominant purple allele. And that's what you saw. They also had the recessive white allele, but you can't see that because there was a purple allele present. And the fourth concept, now known as the law of segregation. So we are going to revisit the law of independent assortment, but here we have another one, the law of segregation. It says that two alleles for a heritable character, so for a particular trait, they separate, they segregate during gamete formation and end up in different gametes. So an egg or sperm gets only one of the two alleles, right, that are present in the somatic cells of an organism. Remember that meiosis produces haploid cells. They have half the total number of chromosomes, which means they have half of the alleles. So only half of them. And the segregation of alleles corresponds to the distribution of the homologous chromosomes, right, to different gametes in meiosis. So remember, in meiosis 1, all the homologous pairs line up at metaphase randomly, and then when the pairs are separated, you get a random mix of chromosomes in those daughter cells. They all have the same number. In humans, that's 23, and they have all the right ones. It's just that they have a mixture of mommy and daddy chromosomes, right, from the original parent of that individual that's producing the gametes. So Mendel's law of segregation occurs during meiosis, where the alleles segregate, and each allele for a trait is packaged into a separate gamete. So if you have big P, right? So this is one of our true breeding parents, all purple. The gametes produced are all big P. In the white, a true breeding parent, it was recessive, so you have two little p's, right? So we're just using p's as our letters to designate the colors here. When we start doing problems and things, there are certain sort of rules you have to follow so everything does not get mixed up, so we can understand what's going on. And so here we just use a big P for the purple characteristic and two little p's for the recessive white characteristic. But our true breeding purple plants, all the gametes have a big P. In the true breeding recessive white pea plants, all the gametes have a little p. When you cross a purple and a white pea plant, you get the hybrids, the F1 hybrids, which have a big P allele and a little p allele. But because they have the big P, all the flowers are purple. But when you take two of your F1 hybrids and you cross them together and make the F2, the F1 hybrids, half the gametes will have a big P and half the gametes will have a little p. Any questions on that? We're going to do a lot with learning how to make the gametes and what the different genotypes of the gametes are. So the law of segregation. And this is kind of a confusing slide because if I ask you which stage of meiosis creates the law of segregation, you're going to think that it's here in meiosis 2 where the sister chromatids split in anaphase 2 and migrate towards opposite poles, right? Because now you're separating all the sister chromatids. But what sets everything up to allow that to happen occurs in metaphase 1. So when you got all those homologous pairs of chromosomes lining up randomly during metaphase, that sets up the process of segregation later. But the point is, remember, all the gametes are not identical to each other. They're all unique. And part of the reason for that uniqueness is the law of segregation, which separates alleles into different gametes. So when we had our big P, little p hybrid, and we said that it can make some big P gametes and some little p gametes, that's because of the law of segregation. You're not going to get a gamete, which is this, right? Because that would be a serious problem. It has an extra chromosome. So those offspring are going to be messed up, and they most likely will not even survive to be born. In this case, we're talking about making seeds, but the seeds are not going to be viable. So we separate the alleles into different gametes, and that's the law of segregation. So we just saw that there was a 3-to-1 ratio in the purple to white flowers. And so segregation helps to account for that 3-to-1 ratio. A way to easily see this is a Punnett square. So you may remember Punnett squares from high school biology. So we are going to be doing lots of those, especially when you work on the genetics problems. And so it's a way to show the genotype of the gametes and how they come together in the possible offspring. So we use capital letters to represent the dominant allele, lowercase letters to represent the recessive allele. So there will be some rules that will follow to keep everything neat and organized. So here we have our true breeding purple flowers, and we cross them with our true breeding white flowers. Since they're true breeding, they're both homozygous for their characteristic. The purple plants are homozygous dominant big P, and the white plants are homozygous recessive, little p. So the purple plants can only produce gametes with a big P allele, because that's all they have, and the white plants only produce gametes with a little p allele. So all the offspring are big P, little p, right? They're hybrids, they're heterozygous. But the big P, little p plants, half of their gametes will have a big P allele, and the other half will have a little p allele, right? Because they're heterozygous, so half the gametes are big P, half the gametes are little p. So when we cross two of the F1s together, we write the gametes on the sides of our square, so the left side and the top side. Whichever side you choose to put the sperm and the eggs, it doesn't really matter. But we put the gametes from one individual on the left and the gametes from the other individual across the top. And then we combine the gametes in the boxes. So a big P sperm and a big P egg, we get big P offspring. A little p sperm and a big P egg, we get a big P, little p offspring. And the same thing here, a little p sperm and a big P sperm and a little p egg, and we get a big P, little p offspring. And then if we have two little p's, eggs and sperm, they come together, and those offspring, and we get the white offspring. Notice that we have a 3-to-1 ratio. And because we're talking about the characteristic that we see, we call that the phenotype, right? That's the observable characteristic. And we have 3 purple and 1 white, a 3-to-1 phenotypic ratio. Now, notice that we put the capital letter first, regardless of whether it comes from the sperm or the egg. And so that's one of our rules, is to put the dominant allele first, followed by the recessive. That helps to keep our table organized. And you'll see this makes a difference when we have a much larger Punnett square to do. Now, when it comes to the genotype, that refers to all the genes, all the alleles, so all the letters that we see. So in the offspring, we have big P, big P, little p, and little p, right? Those are the 3 possible genotypes we see in this table. We have 1 big P. We have 2 big P, little p, right? And we have 1 little p. So that is a 1 to 2 to 1 genotypic ratio. You must know these ratios. These are characteristic of a hybrid cross, right? Because we have a hybrid here and a hybrid here, and it's only one characteristic. In this case, flower color. So whenever you cross 2 monohybrids together, you always get a 3-to-1 phenotypic ratio and a 1 to 2 to 1 genotypic ratio. So those 2 ratios you must memorize. You got to know that for a monohybrid cross. So far, so good. So we've already said that 2 identical alleles means you're homozygous. If they're 2 different alleles, you're heterozygous. heterozygous individuals are not true breeding. Right? We just saw we had two different flower colors, purple and white, right? Even though the hybrid parents are purple, we get some white offspring, so that's not true breeding. And if you, well, most likely not you, but maybe your parents or your grandparents, like to grow flowers in the garden. If you go look at different seed packets, sometimes you'll see on the seed packet, it says F1 hybrid. And you see this especially with things like sunflowers, all these different types of sunflowers they have, they're hybrids. So if you save the seeds, assuming they make seeds, there are some F1 hybrids that do not produce pollen. So they're going to be sterile. They're not going to be able to reproduce and make seeds. But if you can make seeds from your F1 hybrids and then you plant those seeds next year, you don't get sunflower plants that look like the F1 hybrids because they were hybrids to begin with, right? So they're not true breeding. So because of the different effects of dominant and recessive alleles, an organism's traits do not always reveal its genetic composition. So we saw with the big P, little p hybrids, they were all purple. You don't know whether they were homozygous big P, or heterozygous big P, little p, because they're all purple. So there is a way to help determine what the genotype is if you don't know what it is right from your plant. And so we'll see how to do that. But you often cannot tell what the genotype is just by looking at an organism, right? Because they could have the recessive allele, but they've got the dominant one, so that's what you see. But as we'll learn in a future lecture, even if you happen to have a dominant allele, that doesn't necessarily mean you're going to express that characteristic, because it is more complicated than Mendel ever could have imagined. And there's actually some speculation that some of his crosses did not reveal a pattern he could understand. And so he just kind of ignored those and did not include those results in his writings. So he probably did do some things that did not make sense to him and didn't match his previous results. And because he couldn't explain them, he just kind of ignored them. So again, here we see our 3-to-1 phenotypic ratio and our 1-to-2-to-1 genotypic ratio. These are characteristic ratios for a monohybrid cross. So how can we tell the genotype of an individual with a dominant phenotype? So again, our F1 hybrids were all purple, but we don't know if they're homozygous big P or heterozygous big P little p. If you see a pea plant that has a purple flower, you don't know what the genotype is just by looking, right? Because they're all purple. So the way to help determine that is called a test cross. A test cross involves breeding your mystery individual, right? The one with the unknown genotype with a homozygous recessive individual. A homozygous recessive, you know what the genotype is, right? It's got 2 recessive alleles, so you know that. You cross that homozygous recessive with your mystery genotype and look at the offspring and see what you get, and that will help to determine what the genotype of your mystery individual is. So here we have our mystery pea plant. It's purple. It could be big P or big P little p, right? You can't tell by looking because either of these two genotypes are both purple, right? Because they have the dominant purple allele. In the recessive, you know it's homozygous two little p's, so you know that. So by crossing these two individuals together and looking at the colors in the offspring, you can determine what the genotype of the purple flower is. So for example, if the purple flower were homozygous dominant, big P, when you cross that individual with homozygous recessive white, little p, all the offspring are purple. Because if you have two big P's, they appear in all of the offspring. So it doesn't matter what the other allele is. In this case, it's a recessive allele. You see all purple. If, however, your mystery individual was heterozygous, big P, little p, you know you're crossing that with homozygous recessive, little p. If there's a little p in this parent and all little p's in the other parent, you're going to get the little p's coming together in the offspring. So the only way to see the recessive characteristic, both parents must have that recessive allele. So if our purple flower is crossed with a white flower and we get half purple, half white, that means the purple flower was heterozygous, big P, little p. It's the only way to see the white flowers, right, the recessive characteristic, is to have two recessive alleles. Each recessive allele comes from one of the parents. So does that make sense? Because that's very important. Right? The recessive characteristic only shows up in the offspring if both parents are carrying that recessive allele. That's good. So when it comes to the law of segregation, Mendel was just looking at one characteristic, the flower color. The F1 offspring produced were monohybrids, right? They were hybrids, big P, little p. So they were heterozygous. When we cross those two hybrids, we have a monohybrid cross because we're only looking at one characteristic. And so that was our simple, Punnett square. And that's where we get our 3:1 phenotypic ratio and the 1:2:1 genotypic ratio. If we cross two monohybrids together, we always get those ratios in the offspring. So, well, that's pretty easy because it's only one characteristic. What if you have more than one characteristic? Well, Mendel did work with plants that had two characteristics. So he did look at dihybrid crosses. And dihybrids is the most complex we're going to get to in this course. If you go to take genetics, biology 222, you will do some trihybrid crosses. They get very complicated. And beyond that, you really need computers to do that because there's just way too many different alleles for all the different genes. But here we're going to look at two different genes, and each gene has two different alleles. And so the ratios are still characteristic, but they're very different ratios. So here, Mendel looked at color and shape of the pea, right? So the actual seed, the pea. He had yellow and smooth and green and rough. And so for this example, they're using Y's for the color. A capital Y is the yellow, and it's dominant. Lowercase y is the green, it's the recessive. R they're using for the seed coats, right? Smooth and rough, or I guess round and wrinkled is probably what they're using here. Round and wrinkled. So they use R, right, for round, a capital R, and a lowercase r for the wrinkled. So a true breeding yellow round P is mated with a true breeding green wrinkled P. So the green and wrinkle characteristics are both recessive, so you have to be homozygous recessive in order to show those two characteristics. Since we're dealing with true breeding parents, our yellow round seeds are also homozygous, but they're homozygous dominant. So all of the eggs and sperm, remember plant sperm is called pollen, which we're all sneezing with now because a lot of trees are pollinating. So yes, you're inhaling plant sperm when you inhale the pollen. So since this individual is homozygous dominant, all of the gametes, the eggs and sperm, are all big Y, big R, right? That's the only genotype that can exist for the eggs and sperm from this individual. Likewise, this individual, all of its gametes are little y, little r, right? So we cannot get gametes like this, right? That doesn't happen. Law of segregation, right? So we can't have the exact same type of alleles in the gametes, right? So that would mean that they did not segregate. And that would create serious problems, right? They'd have the wrong number of chromosomes. So the individuals produced are dihybrids. Big Y, little y, big R, little r. So they're heterozygous for both characteristics, right? Because if we do a little simple Punnett square, our one gamete is little y, little r, and the other one is big Y, big R. All the offspring are big Y, little y, big R, little r. So notice we always put the dominant characteristic, the capital letter first, but we keep our alleles together. So we put the Y's together, big Y, little y, and the R's together, big R, little r. That keeps things organized. If you start to mix and match, it's going to be impossible to see what the offspring look like. So there are certain rules we have to follow. Right, so capital letters for the dominant characteristic, lowercase for the recessive. Always put the dominant letter first. And in the case of a dihybrid cross, it's combined with whatever the other allele is for that same characteristic. So in this case, yellow color, seed shape. Does that make sense? So what I want to do is actually go through and do this cross and show you. So remember, we're crossing two dihybrids, big Y, little y, big R, little r, with a big Y, little y, and big R, little r, right? Now, when it comes to determining the genotypes of the gametes, do you guys remember, I don't know what grade this was, but remember you learned a math technique called FOIL? Anybody remember FOIL? Nobody remembers FOIL?
Yeah, like first outside and our last.
Right. So it's not really FOIL, but I like to think of it that way because it always reminds me of it. So we're going to look to see how the alleles can come together. And since we're doing two of the same individuals, I'll just do it for one so we can see. So the individual has this genotype, right? So the big Y consort with the big R, right? So that's one gamete. Big Y consort with the little r. That's another possible gamete. Little y consort with the big R, another gamete. And little y consort with little r. So those are the four possible gametes that can be produced by a dihybrid individual. So no other combinations, right, can occur. That would be a serious mistake. So that is how we derive the genotypes of the gametes. So if we make our table, and it's going to be a very big table, and I didn't leave myself much room here, but I'll put on the left one set of gametes. Across the top, it's the same set of gametes because both individuals are dihybrids. Not very evenly divided. So how many possible combinations will there be in this table? $16, right? So that's a lot. But as we'll see, it's not going to be 16 unique phenotypes. But so how would we fill in this table? Well, since the Y's come first in this example, we put the Y's first and the R's. Always put the dominant allele first, followed by whatever the next allele is. So we have a big Y, big R, right? And this one, big Y, big R, little r. Big Y, little y, big R. And this last one is big Y, and that was supposed to be a little y here. Big Y, big R, little r. So here, big Y, big R, little r. Big Y, little r. Big Y, big R, little r. Big Y, little r. Right, so we see right? So that's how we would fill in this table. Notice I kept the Y's together and then the R's together. Always put the capital letter first, followed by whatever the next allele is, either a capital or a lowercase, right? Now, if you look and you start to assign the observable characteristics, what you notice is there's not 16 different phenotypes here, right? Because if you look right here, for example, those are all the same. Big Y, big R, little r, right? So those are all the same. So there are some duplicates. And also when we look at the different characteristics, you're going to see there's actually only 4 phenotypes here. So of all these 16 combinations, only 4 phenotypes. And so if we try and, I don't have the room to write all this out, but when you're solving your next problems, always write everything out, right? So you can see. Here, it's kind of hard to do, but if we look for yellow round, individuals. So in order to be yellow and round, what alleles do you have to have? Right, a capital Y and a capital R. So we have 1 here, right? Yellow round, yellow round, yellow round, yellow round, yellow round, yellow round, yellow round. Yellow round, yellow round. So these are all the combinations that have a capital Y and a capital R. So how many of them did I check? 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's 9 yellow round individuals. I also see yellow wrinkled. And to be yellow and wrinkled, you need what? What genotype do you have to be yellow and wrinkled? You have to have at least 1 capital Y and 2 little r's, right? So 1 capital Y and 2 little r's. So there's here is 1, right? A capital Y and 2 little r's. Here's one that's 2. Here's one that's 3, right? So we have 3. So we got yellows, and then we have green. And we can have green and round. What genotype must you have to be green and round? Two little Y's and a big R, right? Because if you have a capital Y, you're yellow. The only way to be green is to have 2 little Y's, right? To be recessive. And the round is still the dominant characteristic, so you just need to be 1 capital R, right? So 2 little Y's and a capital R. I see 2 little Y's and a capital R here, 2 little Y's and a capital R here, and 2 little Y's and a capital R here. So I see 3 green round. And then the last trait is going to be green and wrinkled. And the only way to be green and wrinkled, right, because we know that green is recessive and wrinkled is recessive. So you have to have both recessive alleles, 2 little Y's and 2 little R's. And there's just one of those. So there's only 4 different phenotypes in this big, huge table. Only 4 different appearances of all the P's. They're either yellow and round, yellow and wrinkled, green and round, or green and wrinkled. So what we have here is another special ratio. 9 to 3 to 3 to 1. That is a phenotypic ratio. The genotypic ratio here doesn't mean anything. But a 9 to 3 to 3 to 1 phenotypic ratio is what you see with a dihybrid cross. So only 4 phenotypes. There's 9 of one, 3 of 2 of the others, and just one lonely phenotype, which is the homozygous recessive phenotype. So 9 to 3 to 3 to 1 is a characteristic phenotype found in a dihybrid cross. So you've learned 3 important ratios here. In a monohybrid cross, you get a 3 to 1 phenotypic ratio, a 1 to 2 to 1 genotypic ratio, and for a dihybrid cross, you have a phenotypic ratio of 9 to 3 to 3 to 1. So 3 very important characteristic ratios that you must know. So questions on how I made this table? You will have genetics problems where you'll do this. But notice the patterns in that table, right? If we go diagonally, you see that set of 4 dihybrids, big Y, little y, big R, little r. That's going to be found in every dihybrid cross you do. Right? That diagonal is there. You always have that one lonely homozygous recessive in the bottom right corner. So there are patterns, right, that I want you to start to observe when you do crosses. So questions? Okay. When I change slides, this is all going to disappear.
So for a cross between 2 trihybrids that will have a different phenotypic ratio than the monohybrids and dihybrids?
Well, no, for a dihybrid cross, you always get a 9 to 3 to 3 to 1 phenotypic ratio for a dihybrid cross. And for a monohybrid cross, you always get a 3 to 1 phenotypic ratio and a 1 to 2 to 1 genotypic ratio, so always. So what we're seeing here is characteristic for whatever future crosses you do. If it's a monohybrid cross, it'll look the same as what we did with the purple and white flowers. We'll just be talking about different characteristics. With a dihybrid cross, it'll look the same as this, except we have different letters for different characteristics. But the end result is going to be the same. So if we jump back to the previous slide, you'll see that it showed all this. So all the different combinations that I did by hand, they're here. And you notice 9 to 3 to 3 to 1 phenotypic ratio. Yellow round, yellow wrinkled, green wrinkled. If you were to actually do a bunch of crosses and count them, they give you some numbers here which approximate what you would see. And again, they're not going to be exact because in the real world, nothing is exact, right? But here they're saying that there were 315 yellow round, 108 yellow green, 101 yellow wrinkled, and 32 green wrinkled. So approximately the same number of the green round and the yellow wrinkled, and about 3 times as many, right, 315 of the yellow round. And if you divide 315 by 32, it's approximately 9. And so you get your 9 to 3 to 3 to 1 ratio. Again, the more pea plants you actually had, if you did this with 5,000 pea plants, you get an almost perfect 9 to 3 to 3 to 1 ratio, because the more offspring you have, the better the numbers are. If you were just to have, say, 20 pea plants in your offspring, you're not going to see a 9 to 3 to 3 to 1 ratio, because there's just too few pea plants there. There's too many other, random variation that can occur, to affect the results. So you need a large number of offspring to see these characteristic ratios. If you have too few offspring, then there are other things that can happen just randomly, which produce different numbers. So you need a lot of offspring to see this. And that's true with any statistical test, right? Has anyone taken any statistics before? If you have or had, you know that sample size matters. If your sample size is too small, your data are unreliable. So you need a large sample size to get a statistically accurate result. So same thing here. The more offspring you have, the closer the numbers will be to the actual characteristic ratios. And we're going to talk a little bit about probability theory here. Just a little, and you don't really have to worry much about it. Because once you start doing the crosses, you'll be able to do some of these probability rules without even thinking about it, right? Because you can see the results in your Punnett square. And so you can automatically look, how many of my offspring of a particular genotype or phenotype do I have relative to the total number of offspring, right? So in reality, this 9 to 3 to 3 to 1, it's 9/16 and 3/16 and 3/16 and 1/16, right? And that gives you 16 out of 16 possible outcomes. So now we're going to revisit the law of independent assortment, which we talked about with meiosis. When the mommy and daddy chromosomes, the homologous pairs, randomly line up, and they end up in different daughter cells, a random mixture, right? Because chromosome number 1 has nothing to do with chromosome number 7, right? So they'll all sort independently, which means that all the sister chromatids will segregate, right, independently as well into the gametes. So we're talking about a pair of alleles, like big P, little p, right? Big P, little p are going to segregate and end up in different gametes. But the big P allele was on one homologous chromosome, and the little p allele was on another homologous chromosome. So again, it's the separation of the homologous chromosomes in metaphase, or they randomly line up, and then in anaphase 1 they're separated. That sets up the process of segregation of the alleles into different gametes. Now, there is something that happens when genes are very close together on a chromosome, right? So the two loci, the two locations, are basically side by side on the chromosome. The closer two genes are together, the less likely a crossover event will occur to separate them. Because remember, crossovers occur with the adjacent sister chromatids, and they switch pieces, and that's a way to mix up the genes, or in this case, to mix up the alleles. But if two genes are so close together on a chromosome, a crossover event will not occur. And so they will not be separated. They will not be segregated. They will always end up together in a gamete. And that's called linkage. And we'll talk a little bit more about linkage in a future lecture. If you go on to take genetics, you'll learn a lot more about linkage. But there are some genes that always sort together. So you always see them together in the offspring. Even though they're two different genes, they're just so close together on a chromosome that they always sort together. So they're not sorting independently, but that only applies to linked genes. So Mendel's second law of heredity is a law of independent assortment. And again, the idea that non-homologous chromosomes align independently. So all those chromosomes 1 through 23, half from mom, half from dad, randomly line up at metaphase. And then they are separated randomly into different daughter cells. So that's how we end up with all the different possible combinations in the gametes. And so we see those again here, right, for our P example. Big Y, big R, big Y, little y, big R, and little y, little r. So the FOIL technique that I showed you, quote unquote for FOIL, that's how you get the genotypes of the gametes. And that's assuming no problems happen, right? You're not supposed to get big Y together in a gamete, or big Y, little y together in the same gamete, right? So that's not right. Something went wrong. So again, big Y, not possible unless there's a serious mistake. Little y, not possible unless there's a serious mistake. Right? Same thing with the other characteristics like the R's here. Right? So you cannot have that happen. Right? The laws of independent assortment and segregation guarantee you'll get that random mix of alleles in the offspring, and we see all the possible combinations, right, that can occur. Nothing else but those 4 combinations can occur from our dihybrid individual, right? So this is it. Unless there's a serious mistake, and we'll talk about mistakes in a future lecture. And so again, independent assortment, which we already knew, happens during metaphase 1 of meiosis 1, where the homologous pairs line up randomly, and then they're separated. Unless they're linked. If they're linked, right, then that means the 2 alleles, right, those 2 genes are so close together on a chromosome, they always sort together, right? So they're not independent of each other, but that's only for linked genes. And we're not going to do anything with linked genes in this course. So let's talk about some probability theory. And some of the stuff that we're going to talk about is not really that important for you to remember. In fact, in the future, I'm thinking about getting rid of some of this information because it's not really necessary for you to understand how to make and use a Punnett square. It's just that it explains why the Punnett square works the way it does. So, with probability, they often use a coin toss as the example. When you toss a coin, there are technically only 2 outcomes when you toss the coin, right? You have heads and tails. Yes, there is that 1/3 improbable outcome, right? Where the coin stands up on the edge, but that's extremely unlikely, so we don't even worry about that probability. So when you toss a coin, you always say there's only 2 outcomes, heads or tails. Each outcome has a 50-50 chance, right? So alleles work the same way, like tossing a coin. So your probability value, your p-value, so p stands for probability, is the number of successful outcomes divided by the number of possible outcomes. So the number of successful outcomes is a number of different ways a specific outcome can occur, and the possible outcomes is simply all the possible outcomes that can occur. Your probability value is always between 0 and 1. It's never greater than 1 because 1 means 100% probability. And you can never have more than 100% probability, right? 100% probability means it's going to happen, right? A 100% chance it's going to happen. 99% chance it's most likely going to happen, but it's possible that it won't, right? So that's the idea behind probabilities. It gives you, or it tells you, your relative chance of an outcome occurring. Like the probability of winning the lotto, right? Winning that huge jackpot. Your probability is extraordinarily small. It's not 0, but it's like you probably 0.001 something. So it's really, really small. So it's possible. It's just extremely unlikely, right, that you'll win the lotto. But let's talk about a deck of cards. If you ever played cards, like, played poker or something, there are 52 cards in a deck. And there are 13 of each suit, right? So the suits are hearts, clubs, spades, and diamonds. What would be the probability of drawing a club from that deck? What would be the probability of drawing a club? Okay, well, you jumped ahead with a math quiz, but we know that there are 13 clubs in that deck of 52 cards. And when we simplify, that's 1/4, or 0.25, or a 25% chance. So there's more than one way to write, the number for the probability. It can be a fraction, it can be a decimal, or you can write it as a whole number percent, right? So there are different ways to express that, but 13 clubs out of 52 possible cards, your probability is 1 out of 4, or 1/4, 0.25, or 25% chance. So what's the probability of drawing a club and a diamond? Of both of them? So we know that there are 52 cards total. We know there are 13 clubs and 13 diamonds, right? So it would be 26 out of 52. And that equals what? Right, so 2 out of 4, and that simplifies to 1 out of 2, or 1/2, which of course can be 0.5. That's a 0.5, or 50%, right? So you have a 50% chance of drawing either a club or a diamond, or a club and a diamond. So if you play poker, right, your chances are not all that good. If you play blackjack, right, 21, your chances are not going to be that good, right? So casinos do not lose money, right? They make money, right? Because your chances of winning any game are low, right? So it does happen occasionally, but casinos always win in the end, right? They win more than they lose. And most people lose more than they win, but they enjoy doing it, right? So we have two rules, the multiplication rule and the addition rule. You don't have to memorize what these mean, because when we start doing Punnett squares, you're just going to do it by looking at the results. But the multiplication rule, also called the product rule, states the probability that 2 or more independent events will occur together is the product of their individual probabilities. So we have at least 2 events, they're independent of each other. So Whatever happens to one has no effect on what will happen to the other. They're independent of each other. And so you multiply the two probabilities together. We're going to look at a fork line diagram to illustrate using heads and tails. But when we draw a Punnett square, what you're actually doing is you're following these rules, but you don't have to think of them in terms of those rules. But if we think of a heterozygous plant like our pea plants and our flower color, and we just have two alleles, either big P or little p, it's like tossing a coin. Half the time you get a big P allele, half the time you get a little p allele. So it's a 50-50 chance in the gametes of getting either a big P or a little p. If you cross two of those F1s together, the monohybrids, then you get different ratios depending upon whether you're looking at the phenotype or the genotype. So here we're going to look at tossing two coins. And so the probability, and this is what they mean by a fork line diagram. Now, in this case, there are technically 4 possible outcomes, because in this example, the order of the outcomes matters. So even though they're independent of each other, heads first, then tails is different than tails first and then heads. So where the order of the outcomes matters, then you see a difference. When it comes to alleles ending up in the offspring, order is irrelevant, right? What's relevant is just what alleles you get, not what order in which you get them, right? Chromosomes are not in order in your cells, right? They're not organized, or here's chromosome number 1 on the left, all the way through to chromosome 23 on the right. Right? That's not how it is inside of a cell, right? So order doesn't matter when you get those alleles in the offspring, but if order does matter, then we see 4 possible outcomes. Each outcome has a probability of 1 out of 4, or 25%. So if the order of the outcomes matters, and if the outcomes are independent of each other and have no effect on each other, you get something like this. But this is not how gametes work, right? This is not how they come together in the offspring. So the addition rule, or the sum rule, says the probability that any one or two or more events, exclusive events, will occur is calculated by adding together the probabilities. So again, we're not going to worry about what the rules are, because you'll intrinsically just know them when you start to do your Punnett squares. And this is what we see with a Punnett square, which is also called a contingency table. Punnett is named after the person, right, who sort of started doing this way. So in this case, when you toss a heads or tails, the order doesn't matter. So heads first, then tails is the exact same as tails first and then heads. Because when it comes to gametes, order is irrelevant. It's just what gametes you end up with, right, that matters. So order makes no difference. If we do the Punnett square, like what we've already done for the peas and the flowers, heads happens 1 out of 4 possible times. Tails happens 1 out of 4 possible times. And a heads and a tails, since they're the same thing, it happens half of the time. So 1/4 plus 1/4, that would simplify to 1/2. So, but this is what you do with a Punnett square. And so when you make your Punnett square, in this example here, we have 4 possible outcomes, right? How often does your outcome occur? Heads, heads occurs 1 out of 4 times. Tails, tails happens 1 out of 4 times. But having both the heads and the tails together, order doesn't matter, happens 2 out of 4 times. And so we get 1/2, or 50%. And so this is what you normally do when you do your Punnett squares, but you're not thinking about any rules. You just look at the table, you look at the number of outcomes in that table, and then what outcome do you want? And remember, it's the outcome that you want divided by the total possible outcomes. And that's just a simple probability. And that's what we normally do with our Punnett squares. So don't worry about multiplication rules or some rules, don't memorize what they are. You'll get the picture when you start doing more Punnett squares. Right, so this is what we normally do, and you do it without even thinking about it, right? So you don't have to know what the rules are in order to actually use them, because once you see the patterns, right, then you get it, you understand, right? Right, so we understand the patterns. So again, when you're doing crosses, monohybrid or dihybrid crosses, you actually incorporate these rules without really thinking about them. So when we did the dihybrid, we added up all the different phenotypes and got our 9:3 to 3:1 ratio in the offspring. Only 4 possible phenotypes, 9 of 1 phenotype, 3 of 2 phenotypes, and 1 unique phenotype. So what we actually did without thinking about it was apply these probability rules, right? But we did that without even knowing what the rules were to start with. So you don't have to memorize the rules. So the relationship between the genotype and phenotype is rarely as simple. As I said, it's a lot more complicated than what Gregor Mendel ever could have imagined. So many heritable characters, right? So many traits are not determined by only one gene with 2 alleles. So as we'll learn later, there are characteristics that have multiple genes and multiple alleles for those genes. And even if you have a particular allele, even if it's a dominant one, you won't necessarily express it. You won't necessarily see it in the phenotype, because there are a lot of other factors that come into play when it comes to gene expression. And we'll talk about those in a future lecture. But the basic principles of segregation, independent assortment do apply to even more complex patterns of inheritance, but there are other things that are going to modify the expression. And we're going to learn about those later on. So I think we're going to stop here for today, because we do have tomorrow to finish this up. So questions, I know it's a lot of stuff.